Hello everyone, welcome to my channel Dogma Biology classes. In this video, our topic is genetic code. This video is taken from chapter 6, part 4 of CBSE class 12th. This is molecular biology series. In previous videos, we discussed about replication, transcription and translation. Now in this video, our topic is genetic code. So at first, what is genetic code? Have you ever written a secret message to one of your friends? If so, you may have used a code to keep the message hidden. For instance, you may have replaced the letters of the word with numbers or symbols following a particular set of rules. In order of your friend to understand the message, they would need to know the code and apply the same set of rules in reverse to decode it. So, decoding message is also a key step in the gene expression, in which information from a gene is read out to build a protein. In this article, we will take a closer look at the genetic code, which allows DNA and RNA sequences to be decoded into the amino acids of a protein. Now, at first, the meaning and role of genetic code. The uniqueness of every cell, individuals or species lies in the uniqueness of its proteins. The cells are enabled to synthesize their specific proteins by the information flowing from the DNA. This information exists as the particular sequence of the bases in the DNA strands and is called genetic code. It is sent to the protein manufacturing machinery in the form of mRNA synthesized on DNA template. The order of bases in the mRNA decides the order of amino acids in the polypeptide to be synthesized. Determination of the sequence of amino acids in a polypeptide by the sequence of nitrogenous bases in the DNA or mRNA is the function of the genetic code. It means that the genetic code is a set of rules by which the information encoded in genetic material and is translated into the protein by the living cells. Now, the nature of genetic code. Here in this picture, you can see the singlet code, doublet code and the triplet code. So, there are 20 types of amino acids and only 4 types of nucleotides, namely adenine, thymine, guanine and cytosine. It is clear that one-to-one correlation between nucleotides and the amino acids is not possible. If one base coded for one amino acid, only 4 amino acids could be specified. That is, the protein could contain only 4 amino acids, like the singlet code. If a sequence of 2 bases, like the doublet code, for one amino acid, the 4 bases could specify 16 amino acids, that is 4 into 4. It means 16 codons. This is also inadequate. If a sequence of 3 bases coded for one amino acid, the 4 bases would specify 64 amino acids, that is 4 into 4 into 4, that is 64 codons. And clearly a sequence of 3 bases is called triplet code, is the simplest code that can account for the 20 amino acids. Singlet and doublet codes are insufficient. The sequence of base triplet in DNA molecules constitute the genetic code. Crick in 1961 concluded that the 3 consecutive nucleotides in mRNA strand determine the position of a single amino acid in a polypeptide chain. Nirenberg and Mathai soon provided experimental evidences to show that genetic code is a triplet code. So this is the nature. Now the details of the genetic code. Here you can see the standard genetic code, the sequence of nucleotides in the triplet codons of the RNA and each of which specifies a particular amino acids. The first codon was deciphered in 1961 by Marshall Nirenberg. It was UUU for phenylalanine. Marshall Nirenberg and Harbind Khorana have determined which sequence of bases coded for which amino acid with the help of experiments. They received a Nobel Prize for this work in 1968. The genetic code enumerated by them is given in the table you can see here, and this table shows codes in terms of messenger RNA and not of DNA. The reason for this practice is that the cell reads the code from messenger RNA molecule and not directly from the DNA of chromosomes. The mRNA is read from the fifth end towards the third end. Now, in this picture you can see the genetic code. Cracking the genetic code, the language of DNA required determining how the four letters could encode more than 20 different amino acids. Francis Crick and Sidney Brenner determined that the DNA is read in sets of three nitrogenous bases called a triplet. In RNA, it is called a codon. Scientists identified that the triplet codes for a specific amino acids and that a specific amino acid codes for a certain proteins. This is the genetic code table. Here, full set of relationship between the codons and amino acids is called genetic code. The genetic code is often summarized in a table. And how do you read the codon table? So for this, the codon table may look kind of intimidating at first. Fortunately, it's organized in a logical way and it's not too hard to use once you understand this organization. To see how the codon table works, let's walk through an example. Suppose that we are interested in the codon CAG and want to know which amino acid it specifies. First, we look at the left side of the table. The axis on the left side refers to the first letter of the codon. So we find C along the left axis. This tells us the row of the table in which our codon will be found. Next, we look at the top of the table. The upper axis refers to the second letters of the codon. So we find A along the upper axis. This tells that the column of the table in which our codon will be found. So the row and column from step first and second intersect in a single box in the codon table, one containing 4 codons. It's often consists to simple look at these 4 codons and see which one is the one you're looking for. So if you want to use the structure of the table to the maximum, however, you can use the third axis corresponding to the intersect box. By finding the third nucleotide of the codon on this axis, you can identify the exact row within the box where your codon is found. For instance, if we look for G on this axis in our example above, we find that CAG encodes for the amino acid glutamine. So this is all about that how do you read the codon table. Now, the characteristics of the genetic code. And if you talk about the characteristic, the first character is triplet nature. The genetic code is a triplet code and the 3 adjacent bases, that is codon, specifies one amino acid. Second is non-overlapping. The adjacent codon do not overlap, they do not share any base. So the sequence, that is AUU, UUG, UGC, is read only as AUU, UUG and UGC. The code for a protein containing 100 amino acids unit consists of 300 nucleotides linked in a linear order. If the triplet code involving overlapping, a series of 6 nucleotides could code for at least 4 amino acids. This is not the case. So here the codes, that is AUU, UAG and UGA, codes for isoleucine, leucine and cysteine amino acids. Next is no punctuation. The genetic code is commaless. Next is universality. The genetic code is universal. That is, a given codon in the DNA and mRNA specifies the same amino acid in the protein synthesizing system of all organisms from bacteria to man, also in viruses. Next is degeneracy, that is redundancy. The genetic code is degenerate, that is, it lacks specificity and one amino acid often has more than one code triplet. Only methionine and tryptophan has a single triplet codon. So the genetic code is redundant, also called degenerate, although each codon corresponds to a single amino acid, but a single amino acid can have multiple codons except tryptophan and methionine. Each amino acid has a multiple codons. Next is non-ambiguity of codons. Each codon codes for only one amino acid, none for more than one. No doubt, the codons GAA and GAG both specify glutamic acid. Neither of them specifies any other amino acids. Next is nonsense or terminator codon. So, there are 3 of the 64 codons, that is UAA, UAG and UGA, do not specify any amino acid but signal the end of the message. They are called the nonsense or terminator codons. Next is initiation of start codon. The codon AUG is called the initiation or start codon as it begins the synthesis of polypeptide chain. Next is collinearity. DNA is a linear polynucleotide chain and the protein is a linear polypeptide chain. The sequence of amino acids in a polypeptide chain corresponds to the sequence of nucleotide bases in the gene that codes for it. Changes in a specific codon in the DNA produces a change of amino acid in the corresponding position in the polypeptide. The gene and the polypeptide it codes for are said to be collinear. Next is gene polypeptide parity. A specific gene transcribes a specific mRNA which produces a specific polypeptide. On this basis, a cell can have only as many types of polypeptide as it has types of genes, and this does not apply to certain viruses which have overlapping genes. So this is all about the characteristics of the genetic code. Now, the effect of mutation on protein structure and function. At first, the changes in the protein structure. Protein structure is altered by 3 types of mutation. First one is gene or point mutation, which involve addition, deletion or replacement of a base pair in a gene. Second is addition or deletion of DNA chunks during crossing over between DNA strands under special condition. And the third is jumping genes, which shuffle their location from one chromosome to another due to similarity of the DNA sequence flanking them. Now, the effect of altered proteins. All the above mutations are reflected in the proteins. So, this is because the mRNA transcribed from the mutated gene will lack a segment or a base or will have an additional or altered segment or base. The abnormal mRNA will introduce different amino acids in the polypeptide synthesized with its altered message. If a single base is altered in the gene, only one codon in mRNA will change, and this will change a single amino acid in the polypeptide. If the number of bases inserted in or deleted from a gene is not divisible by 3, all the base triplet beyond the site of mutation are altered in the mRNA transcribed from it. So, this will result in the formation of entirely a new polypeptide, and this is called frame-shift mutation. Mutation may change a normal codon to a nonsense codon. If so, protein synthesis will stop at this codon and an incomplete polypeptide will be formed. Now, WOBBLE hypothesis. So, what is WOBBLE hypothesis? A change in a single base may not change the amino acid in the polypeptide. This happens when the third base in a triplet is changed because such as codon can still interact with the anticodon of the corresponding tRNA. This phenomenon was called WOBBLE hypothesis by Crick in 1966. So, WOBBLE in the anticodon. In 1966, Francis Crick proposed the WOBBLE hypothesis. According to this hypothesis, only the first 2 bases of the codon have a precise pairing with the bases of the anticodon of mRNA, while the pairing between the third bases of the codon and anticodon may WOBBLE, that is non-specific. The pairing between the first 2 bases is normal, while between G and U is against the normal pairing patterns, And this unusual bonding between the G and U is called WOBBLE pairing. Like inosine is the fifth base in the anticodon. Inosine arises through enzymatic modifications of adenine by ADAR, adenosine deaminase RNA specific. Inosine is commonly found in tRNAs and is especially for the proper translation of the genetic code in WOBBLE base pairs. Advantage of WOBBLE is dissociation of tRNA from the mRNA is faster. So Crick proposed the WOBBLE hypothesis to explain the degeneration of the genetic code. Except the tryptophan and methionine, more than one codons direct the synthesis of one amino acid. There are 61 codons that synthesizes amino acid, therefore there must be 61 tRNAs, each having different anticodons, but the total number of tRNA is less than 61. So the human disorders called the sickle cell anemia is caused by a single base substitution in a gene, and this alteration results in the replacement of glutamic acid by the valine at position 6 in the beta polypeptide chain of the hemoglobin, making the letter hemoglobin S. The disorder is manifested only in the homozygous recessive state. It kills the victim by the age of 20 years. Thalassemia is also another genetically transmitted hemoglobin disorder. So this is all about the WOBBLE hypothesis. Next is the regulation of gene expression. So if we talk about the regulation, the phenomena of gene repression and depression provides a means of decreasing or increasing the rate of synthesis of a particular enzyme in response to variations in the environmental requirements for the enzyme. In other words, a cell has a mechanism that controls the expression of a particular gene and thereby the amount of a particular enzyme at a particular time. The rate of protein synthesis is regulated by genetic apparatus and environmental factors in accordance with the need of a cell. The mechanism which stimulates the expression of certain genes and inhibits the expression of others in the cell is called control or regulation of gene expressions. Now, the regulation of gene expression in case of prokaryotes. And here, the lactose or lac operon model for the gene regulation in prokaryotes. Bacteria can regulate the synthesis of enzymes in such a way that the enzymes are produced only when the substrate on which these enzymes have to act are present. In 1961, two French microbiologists, Francois Jacob and Jacques Monod, at the Pasteur Institute, Paris, proposed a mechanism called operon hypothesis for the regulation of gene action as a result of their studies on the metabolism in E. coli. Operon is a segment of DNA that comprises one or more adjacent structural genes, an operator gene, a promoter gene, and a regulator gene. Operon works in cooperation with the repressor proteins and an inducer substance. An operator acts as a gene regulating unit. So this is all about an introduction of the lactose or lac operon model. After this, now the types of operon. And here operons are of two types, in which the first one is inducible operon. Inducible operon, here you can see this is lac operon, which is an inducible operon. Normally the proteins needed to break down lactose are turned off because there is a glucose around for the bacteria to use. It is not a simple on and off switch, it is more like a light demo. This operon is inducible because it is normally off. If glucose and lactose are both present, the operon will still be turned off. So, the lac operon of E. coli contains genes involved in lactose metabolism. It is expressed only when lactose is present and glucose is absent. Two regulators turn the operon on and off in response to lactose and glucose levels that are lac repressors and CAP, that is catabolic act here. that is activator protein, CAP, catabolic activator protein. So the lac repressor acts as a lactose sensor and it normally blocks transcription of operon but stops acting as a repressor when lactose is present. The lac repressor senses lactose indirectly through its isomer alactose, and CAP acts as a glucose sensor and it activates transcription of the operon, but only when the glucose levels are low. CAP senses glucose indirectly through the hunger signal molecules, that is CAMP. So, the operon model proposes three elements: a set of structural genes, that genes encoding the proteins to be regulated, an operator site, which is a DNA sequence that regulates the transcription of the structural gene, and the regulator gene, which encodes a protein that recognizes the operator sequence. Lac operon is one of the most studied operon of the E. coli. This codes for key enzymes involved in lactose metabolism. That is galactose permease. First enzyme is galactose permease or lactose permease. It transports the lactose into the cell across the membrane. Second is galactosidase, which hydrolyzes lactose to glucose and galactose. And the last is thiogalactoside transacetylase. Now, in the lac operon, the structural genes are LAC-Z, LAC-Y and LAC-A, encoding galactosidase, permease and transacetylase respectively. Transcription occurs from a single promoter that lies upstream of these structural genes and binds RNA polymerase. However, also present are an operator site between the promoter and the structural genes and a LAC-I gene that codes for the lac repressor protein. The LAC-I gene has its own promoter that binds RNA polymerase and leads to transcription of lac repressor mRNA and hence the production of lac repressor protein monomers. Four identical repressors monomers come together to form the active tetramer, which can bind tightly to the lac operator site, that is O-lac. Now, inducers and the inductions of the lac operon. Normally, E. coli cells make very little of any of these three proteins, but when lactose is available, it causes a large and coordinated increase in the amount of each enzyme. Thus, each enzyme is an inducible enzyme and the process is called induction. The mechanism is that the few molecules of beta-galactosidase in the cell before induction convert the lactose to allolactose, which then turns on the transcription of these three genes in the lac operon. Then allolactose is an inducer. Another inducer of the lac operon is propylthiogalactoside. So here, unlike the allolactose, this inducer is not metabolized by the E. coli and so is useful for the experimental studies of the induction only. Now, here you can see that the absence of lactose and in the presence of lactose. What is inducible operon here? So, at first, in the absence of lactose, the regulator protein binds to the operator and inhibits transcription. But in the presence of lactose, when lactose is present, some of it is converted into allolactose, which then binds to the regulator protein, making the protein active. Then the regulator protein cannot bind to the operator and the structural genes are transcribed and translated. So this is all about the lac operon model. Here you can see that the lac operon and its control elements. So the inducible operons generally function in catabolic pathway. Here you can see that the LAC-Z, LAC-Y and LAC-A genes form the enzymes, that is messenger RNA. When the low glucose and lactose available, In this case, lac genes is strongly expressed. When the high glucose lactose unavailable, lac genes not expressed. When the low glucose lactose unavailable, lac genes not expressed. And when the high glucose lactose available, very low, that means basal level of gene expression occur. So this is all about the lac operon and its control element. Now the next, that is repressible operon. And if we talk about the repressible operon, the operon is switched off when a chemical substance called co-repressor is present. So the tryptophan or TRP operon of the E. coli is an example of the repressible operon, and it has the following components. Now you can see the structural genes, then operator genes, then promoter genes and regulator genes for the role of repressible operon. So at first, structural genes. E. coli synthesizes tryptophan from a precursor molecule in a series of 5 steps, each catalyzed by a specific enzymes. 5 genes codes for these enzymes, and these genes transcribe mRNA, which are translated by the ribosomes and tRNAs into the polypeptides. The latter function as enzymes. The tryptophan operons comprises 5 structural genes denoted as TRPE, TRPD, TRPC, TRPB and TRPA that lie together on a chromosome. Advantage of grouping the functionally related genes into one transcription unit is that a single on-off switch can control all of them. Now, here, at first you can see that is the operator genes. These genes control a specific series of structural genes and lies adjacent to them. It remains turned on because it is not joined by the aporepressor protein produced by the regulator gene. Therefore, the cistron keeps functioning, producing enzymes. The latter catalyze the synthesis of tryptophan from the raw material. Here, the product of regulatory gene activates the structural genes to carry on transcription. This is called positive control. And the next is promoter gene. These genes lie adjacent to the operator gene. It marks the site where the RNA polymerase enzyme binds. When the operator gene is turned on, this enzyme moves over the operator gene to the structural genes and transcription starts. Now, the next is regulator genes. It controls the operator genes in cooperation with a substance called co-repressor. A co-repressor may be the end product of the metabolism accumulated in the expressed in the cells or the received by the cell from outside. It is tryptophan in the operon being discussed. The regulator genes codes for a protein named aporepressor. The latter combines with the co-repressor, and the aporepressor co-repressor complex acts as a repressor and joins the operator genes, which is turned off. The structural genes now stop transcription and enzyme synthesis ends. So, in case of TRP operon, in case of inactive repressor, polypeptide that makes of enzymes for the tryptophan synthesis occur. But when the active repressor, then the polypeptide synthesis will not occur. So when the organism gets tryptophan in excess from the outside, it conserves its raw material and energy by suspending the synthesis of this compound in its own body. This phenomenon is called feedback repression. So the repressible operon generally functions in the anabolic pathway. Now you can see here the high level of tryptophan and low level of tryptophan. In case of high level of tryptophan, complete ladder of polypeptide form, while in low level of tryptophan, incomplete ladder of polypeptide form. So the co-repressor is a non-protein compound which may come from the outside or from metabolic within the cell. Its concentration controls the transcription in the cell. If concentration of a co-repressor rises, aporepressor co-repressor complex is formed, and the complex joins the operator gene and the further production of co-repressor stops. If the co-repressor level falls, aporepressor fails to get co-repressor and does not join the operator gene. This restarts the formation of co-repressor. So the end product is generally utilized in some other metabolic pathway so that it does not accumulate and fails to act as a co-repressor. If somehow it accumulates or is received from outside, it combines with an aporepressor and stops transcription. So this is all about the TRP or tryptophan operon, that is repressible operon. Now here a comparison between the inducible and repressible operon. So there are some features on which we will distinguish between inducible and repressible. At first, the regulator protein, that is negative state. In case of inducible, active, binds to operator, while in case of repressible, inactive, does not bind to operator. Second is regulator protein after association with effector. In case of inducible, becomes inactive, while in case of repressible, becomes active. Next feature is transcription of the operon in the presence of effector. Inducible operon occur while the repressible operon does not occur. Next is effector termed as inducible operon inducer while in repressible operon co-repressor. Next is active operon is termed as induced in inducible operon and depressed in repressible operon. An operon that cannot become active is called non-inducible in inducible operon and super repressed in repressible operon. So this is comparison. So what are the main differences between the induction and repression? First, induction turns the operation on and repression turns the operon off. Induction starts transcription and protein synthesis while the repression stops transcription and protein synthesis. Induction is caused by a new metabolite which needs enzymes to get metabolized. Repression is caused by an excess of existing metabolite. Induction operates in a catabolic pathway while the repression operates in an anabolic pathway. Repressor is prevented by the inducer from joining the operator gene, and here aporepressor is enabled by a co-repressor to join the operator gene. So this is all about the differences and comparison between the induction and repression and inducible operon and repressible operon. Now, what is gene expression? Gene expression is the process by which the information encoded in gene is used to direct the assembly of a protein molecule. What is regulation of gene expression? Regulation of gene expression includes a wide range of mechanisms that are used by cells to increase or decrease the production of specific gene products, that is protein or RNA. Now, the regulation of gene action in case of eukaryotes. In eukaryotes, the genes that code for the enzymes needed to catalyze the various steps of a particular metabolic pathway may not be adjacent or even be in different chromosomes. However, these genes are regulated by operon model as in bacteria. Both the inducible and repressible system of the operon model work in eukaryotic cells but through a complex network of regulatory genes. The following points are notable about the gene regulation in eukaryotes. First one, that is the various structural genes involved in a particular metabolic pathway are often not located adjacent to one another. They usually occur far apart on the same or even different chromosomes. The structural genes having coding and non-coding segments called exons and introns respectively. Each structural gene appears to have its own promoter gene. There are sensor genes to pick up changes in the intracellular environment such as presence or lack of a substance. There are integrator genes to coordinate functioning of structural genes located in different parts of the DNA. There are enhancer and silencer genes which accelerate or retard transcription. So, here you can see the levels of regulation of gene expression. At first, when the gene or DNA converted into the primary transcript, at that time transcriptional control. When the primary transcript formed the mRNA, at that time RNA processing control. When the mRNA forms the protein, Then in this time, RNA transport control and the translation control. So the control of gene expression in case of eukaryotic cells on transcriptional level, on RNA processing level, on translation level, on protein activity control level. So now the coordination of the histones and non-histones in gene regulation in case of eukaryotes. Here at first the chromatin condensed inactive is converted into the chromatin inaccessible to RNA polymerase in presence of histone crosslinks. So in case of eukaryotes, the gene regulation is also thought to depend on histones and non-histones proteins associated with the DNA. The histones have a general non-specific control that does not distinguish between the individual genes. They, in their normal form, inhibit transcription modification of the histones that makes large blocks of the gene available for the RNA transcription by loosening the tightly packed inactive chromatin. The specific non-histone proteins then determine which of the available genes are to be actually transcribed. For this, the non-histones recognizes a specific DNA sequence and combines with the DNA or its histones to open up the chromatin sufficiently for the RNA polymerase enzymes to reach the DNA. The appearance of specific non-histones regulator proteins at certain times might be controlled by the factors such as hormones or other chemical signals acting on the cell. So this is the coordination of histones and non-histones in gene regulation in case of eukaryotes. Now, the level of regulation in the bacterial gene expression. So in case of gene expression in bacteria can be regulated at three levels. First one is the transcriptional level, then translational level, and after that post-translational control. So this is here the level, the mode of regulation that is the slowest in response time and that which is fastest. So the level the most efficient and least efficient in resource use. So this is all about the level of regulation in the bacterial gene expression at transcriptional level, at translational level and at post-translational level. So it means when the DNA forms the RNA at that time, when the RNA forms the protein at that time, and after the protein formation, that means in the process of modification, you can control or regulate the gene expression. Now, the control of gene expression. If we talk about at the transcriptional control, controls when and how often a gene is transcribed. Next is post-transcriptional control. When the RNA processing control determines how RNA is processed. RNA transport and localization control, selection of the mRNA to be transported to the cytosol. And the last is mRNA degradation control, when the selection of which mRNA to be destabilized. Next is translational control, selection of which mRNA to be translated. And next is protein activity control or the post-translational control, where the regulation of proteins by the activation, deactivation, degradation, compartmentalization and modification of proteins. So these are the steps on which the control of gene expression occur. Now, the advantages of the regulation of gene action. So three main advantages occur for the gene regulation, in which the first one is an operon is a unit of transcription. It is not economical for the cell to use up its energy and materials in the synthesis of enzymes that are not immediately needed. Hence, the protein synthesis is stopped. Next is gene regulation enables the cells to adjust their metabolism to environmental changes. Next, gene regulation also plays a role in growth and differentiation, causing morphogenesis. And the last is the constitutive genes keep on functioning all the time in all cell types and they need not be repressed. Therefore, the regulator and operator genes are not associated with them. So these are the advantages of the regulation of gene action. Now, the housekeeping genes. It has been noted that in case of bacteria, the gene expression is regulated at cellular levels to meet the demand of the environment. In multicellular organisms, there is gene regulation at tissue level also. For instance, some genes express in the liver to produce bile, others express in the muscles to synthesize contractile proteins, still other expresses in the nervous tissues to form neurotransmitters and so on. So, genes that are expressed in certain tissues only are called the tissue-specific genes. In contrast to these genes having limited expression, there are set of genes which are expressed in all the cell types and almost all the time. Such genes are called housekeeping genes or constitutive genes. The product